All right, we're going to discuss now section 6 of chapter 4 with cellular structures. You should be able, after going over this section, list the membrane-bound organelles of a typical human cell, describe the structure and main functions of each, list the non-membrane-bound organelles of a typical human cell, and describe the structure and function and main function of each. Distinguish between cilia and flagella in terms of both structure and function. Describe the structure and function of microvilli, and compare and contrast the structure and function of three major types of membrane junctions. So starting with the membrane-bound organelles, they're surrounded by a membrane, which allows activities in an isolated environment. The endoplasmic reticulum is an extensive interconnected membrane network. It varies in shape, but it's one continuous lumen. It extends from the nuclear envelope to the plasma membrane. It composes about half the membrane within a cell, and there are points of attachment for ribosomes. With ribosomes, the rough endoplasmic reticulum, and without ribosomes, it's a smooth endoplasmic reticulum. So we can have a little picture here, be able to see the nucleus and the cisternae of the organelle. You see a transport vesicle here, the peroxisome, which we'll talk about in a bit here. And here's the rough endoplasmic reticulum and smooth endoplasmic reticulum. You can see through a TEM image here, seeing that rough endoplasmic reticulum and the ribosomes. As far as the functions of the rough endoplasmic reticulum, it synthesizes proteins for secretion, incorporation in the plasma membrane, and as enzymes within those lysosomes. It processes the molecules, modifying the proteins, adding carb to form glycoproteins and tags for shipping and store proteins. Its organelle formation helps forming the peroxisomes, and vesicle formation helps form and transport vesicles for shipping of the proteins to the Golgi apparatus. As far as functions of smooth endoplasmic reticulum, synthesis the site of lipids, steroids, and their synthesis. It processes molecules like carb metabolism, glycogen synthesis, detoxification. It detoxifies drugs and poisons, and vesicle formation, forming transport vesicles for shipping to Golgi apparatus. So a lot going on there. As far as the rough ER goes then, as just mentioned, production by the ribosomes inserted into the endoplasmic reticulum. The original structure of the protein is unchanged, and they're transported out in closed membrane sacs through these transport vesicles, shuttle the proteins, the rough ER lumen to the Golgi apparatus. Smooth ER has diverse metabolic processes that vary by cell. Functions include synthesis, transport, and storage of lipids, carb metabolism, and detoxification of drugs and poisons. As far as the Golgi apparatus, that's composed of the cisternae, elongated sac-like membranous structures. They exhibit polarity, with their cis face being proximal to the ER, to the endoplasmic reticulum, and the trans face is distal from the endoplasmic reticulum. And that said, their functions are modification, packaging, and sorting of proteins, the formation of secretory vesicles. Some vesicles become part of the plasma membrane, and others release contents outside the cell. So here, looking at our picture, see another TEM image of the cisternae, this time of the cisternae. As far as synthesis here, the Golgi apparatus forms proteoglycans, and they process the molecules that modify and storing protein that was formed by the rough endoplasmic reticulum. And the organelle formation, synthesizing digestive enzymes for lysosomes. Vesicle formation, forming secretory vesicles for delivering components of the plasma membrane and releasing contents from the cell by exocytosis. You can see the cis face here, and then the trans face over here. The secretory vesicle is working there, and a better overall picture of what's going on here. Seeing here where the rough ER synthesizes protein that is released into transport vesicles, and the vesicle from the rough ER then transports or moves to the Golgi apparatus. Okay. And then the vesicle here fuses with the Golgi apparatus and at the cis face. And the proteins are modified, moving through here, the Golgi apparatus. They're modified as they move through it, like the addition of carbs. Modified proteins are packaged here, referring to the reference here, with the plasma protein to either insert molecules into the plasma membrane or to release contents via exocytosis, or vesicles also provide digestive enzymes to the lysosomes. The lysosomes themselves are small membranous sacs. Okay, you can see the TEM image here. Contains the digestive enzymes formed by the Golgi. They participate in digestion of unneeded substances, stuff you don't need. And that said, they digest contents of endocytosed vesicles as well. As far as the peroxisomes go, they are membrane-enclosed sacs that are smaller than lysosomes. They're pinched-off vesicles from the rough ER. Their proteins are incorporated to serve as their enzymes, and they include roles in chemical digestion, in beta oxidation, and lipid synthesis, helping form lipids. The clinical view, these points will be here once in a while. If we have time when the content is not so significant or extensive, I will go over them, but otherwise it'd be more reference more toward AP2 really, but they're here for you getting a little bit ahead, so to speak, on forming kind of a mastery level of understanding of the information. As far as the membrane-bound organelles go, the endomembrane system, it's an extensive array of membrane-bound structures. It includes the ER, the Golgi apparatus, vesicles, lysosomes, and peroxisomes. It includes the plasma membrane, the nuclear envelope, and it connected directly or through the vesicles, moving between them. And it provides a means of transporting substances within the cells. Now the mitochondria, which we've mentioned before, of course, are oblong-shaped organelles with a double membrane. Okay. They're used particularly with aerobic cellular respiration. That's primary purpose. And they complete digestion of fuel molecules to synthesize ATP that the powerhouse of the cell. The ribosomes contain the protein and ribonucleic acid. They're arranged into large and small subunits. The large subunit with AP and E sites, the exit site, the peptidal, and the aminoacyl sites. They're made within the nucleolus and assembled in the cytoplasm. They're bound ribosomes attached to the external surface of the ER membrane. They synthesize proteins for export. They become part of the plasma membrane, and they serve as enzymes within lysosomes. Whereas free ribosomes are suspended within the cytosol itself. All the proteins within the cell are synthesized here. So here we can see the ribosomes, the large subunit, the EPA there, forming the small subunit to form the functional ribosome. And the centrosome is a pair of perpendicularly oriented cylindrical centrioles that's surrounded by the amorphous protein. Their primary function is to organize microtubules within the cytoskeleton, and they function in cellular division. And so we can see the centrosome here, the longitudinal section, and the microtubule here with that centrosome, little centrioles here. Okay. Now, proteasomes are large barrel-shaped protein complexes that digest organelles. They are protein digesting organelles, I should say. They're located in the cytosol and the cell nucleus. They degrade cell proteins through ATP-dependent pathways. And they, for example, like damaged proteins, incorrectly folded proteins, as we talked about before, you incorrectly fold the protein, you damage that protein, you can't make use of it, or proteins that are no longer needed. Proteins that are marked with ubiquitin tag for disposal, with age that may be unable to normally remove proteins. So here's a proteasome, seeing their basic structure here, and the ubiquitin here, with protein to be graded is first tagged with the ubiquitin molecule so that it can be recognized. Okay. Here's the amino acids and the peptide fragments that are released to be recycled by the cell. Cytoskeleton plays roles in intracellular support, organization of organelles, cell division, movement of materials. It extends throughout the cell interior and anchors proteins in the membrane. It includes the microfilaments, the intermediate filaments, and the microtubules. The microfilaments are the smallest, not surprising, components of the cytoskeleton. The actin protein monomers are involved in two twisted filaments, and their functions are maintain cell shape, internal support, and cell division. The intermediate filaments are intermediate size, they're more rigid than the microfilaments, and their function is to provide structural support, which makes sense with the rigidity, and cell junctions. And of course, the microtubules are the largest components of the cytoskeleton, composed of tubulin. They can be elongated or shortened as needed. Their functions are to maintain the shape, cell transport, and cell division. So we get a closer look here. The mitochondrion and the centrosome here. And you see the microfilaments there, the microtubules here in blue, and intermediate filaments here in brown. Okay. And that said, or actually showing more over here and over here as well, but brown there. And the functions of that cytoskeleton again, which we mentioned already here. The cilia are hair-like projections that move substances along the cell surface. The flagella is longer and wider than the cilia, propels the entire cell, and the microvilli are extensions of the plasma membrane that increase the surface area. Okay. So here's the microvilli, attach those microfilaments. The membrane junctions are tight junctions. They're strands or rows of protein-like linking cells. They prevent substances from passing between the cells. Makes sense, having a tight junction, making less room, right? Requires materials to move through rather than between cells. Desmosomes are composed of proteins that bind the neighboring cells, such as the hemidesmosomes, okay, hemidesmosomes, which anchor the basal layer of the cells of the epidermis to underlying components. The gap junctions are formed these tiny fluid-filled tunnels, provide direct passageway for substances to travel between the cells, like the ions between cells in cardiac muscle. Here we have the membrane junction here. We can see a tight junction here, membrane protein, the plasma membrane, the microfilaments here, intracellular space, adjacent plasma membranes. Here we have hemidesmosomes, whereby seeing over here like a desmosome, the protein filaments, the protein plaque, a little more space through here, the intracellular space, and the gap junctions with their pore and their connexin. You should be able to, with this section, then describe the general structure of both the endoplasmic reticulum and the Golgi apparatus, discuss their functional relationship in the synthesis, modification, storage, and release of molecules. Lysosomes and peroxisomes are small, with their membrane closed sacs, but which of these functions is involved in both digestion and the synthesis of molecules, let alone the digestion of unwanted organelles. Which non-membrane-bound organelle functions to digest unwanted proteins, form the structural support of the cell, synthesize proteins, and participate in cell division. And then which cellular surface structure functions in increasing cell surface area and moving material past the cell. And of course, which cellular junction provides resistance to mechanical stress, allowing the passage of ions between cells and protein leakage between cells. And that's section 4.6, and then we'll talk about section 4.7 shortly.